Hello everybody. So right till now we have discussed about the introduction of the cell, and we were trying to see what are the structural organizations of the cell. And you have seen that every cell is basically having three basic components, and the three basic components are the cell membrane or plasma membrane, which is the outermost covering of the cell, the nucleus and the cytoplasm. So we have seen that plasma membrane, which is selectively permeable, which is flexible, it provides a flexibility to the cell, and it is also responsible for controlling the entry and exit of various materials across the cell membrane. But if we talk about the plant cells have got one more additional covering, and that more additional covering is known as cell wall. But remember, this cell wall is found only in plant cells, not in animal cells, right? So plant cells are having that additional covering. You have seen in the cell, the outermost covering is plasma membrane. Inside the plasma membrane there are the various cell organelles about which we will be discussing later on. So but remember that the cell is having the outermost covering, that is cell membrane or plasma membrane. But if you talk about the plant cells have an additional covering around the cell membrane. And that additional covering found in plant cells is known as what? It is known as cell wall. So as you have already seen that cell membrane or plasma membrane is made up of lipids and proteins that are combinedly known as lipoproteins. But if we talk about the cell wall is made up of cellulose, which is a complex carbohydrate. Not only plant cells, but you can say fungal cells, means the cells of fungus. Fungal cells are also having cell wall, but their cell wall is made up of chitin, which is a complex sugar, right? So in this way we can have the cell wall only in plant cells, and that cell wall is made up of what? It is made up of cellulose. But in fungus cell there is a cell wall, and this cell wall is made up of chitin, right? If we look at the functions and characteristics of the cell wall, as this cell wall is rigid, so it provides a definite shape to plant cell. So due to presence of the rigid cell wall, plant cells cannot change their shape, right? Another important property of that cell wall is this cell wall is highly permeable. That means it allows each and every material to pass through it, whereas you have seen that cell membrane or plasma membrane was selectively permeable, but this cell wall is highly permeable, right? Another important property is that it protects the plant cell from getting burst in hypertonic solution. right? So for example, if we consider the case of an animal cell, suppose here we have an animal cell kept in hypertonic solution. So as we have already discussed in previous lecture, that when a cell is kept, an animal cell is kept in a hypertonic solution, what happens? The water moves from outside to inside, and the process occurring here is endoosmosis, and due to which the cell swells. And if this endoosmosis keeps on uncontrollably, then what will happen? The water will keep on entering, the cell will keep on swelling, and this water will exert a pressure from inside to outside on the cell membrane, and ultimately a time will come when the cell membrane will burst, right? Exactly in the same way, like if you take a balloon and you keep on filling water to this balloon, what happens? As soon as you fill the water, this balloon expands. You keep on filling the water, the balloon keeps on expanding, but there is a certain limit up to which this balloon can expand. And that happens, that when excessive water is there, what happens? The balloon will burst. Exactly in the same way, the animal cell also burst, right? But instead of that animal cell, if we would have kept a plant cell, like we have kept this plant cell in the same hypertonic solution, then what will happen? Here also, initially the endoosmosis will occur and the water will enter the cell. Now this water will exert a pressure on cell wall membrane from inside to outside. At the same time, the cell wall exerts an equal pressure on the cell membrane from outside to inside, right? So the pressure exerted by the water on the cell membrane from inside to outside is counterbalanced by the pressure exerted by cell wall on the cell membrane from outside to inside. So the two opposite pressures cancelling out each other, and the net result is your cell is protected against bursting. So that's what I was telling, that a cell wall protects the plant cell from getting burst when it is kept in a hypertonic solution, right? So another very important application or function of you can say cell wall found in plant cell. Another important function which a cell wall in the plant cell performs is there are the processes known as plasmolysis and D Plasmolysis. Again, in order to understand the process of plasmolysis or deplasmolysis, let's consider a plant cell. Now this time the plant cell is kept in a hypertonic solution, right? So here we have a hypertonic solution. In this hypertonic solution, we have kept a plant cell which is having the outer rigid covering of cell wall, right? So now you know, whenever a living cell is kept in hypertonic solution, what happens? The process occurring here is the flow of water from inside to outside, and that process which occurs here is known as exoosmosis. And as you know, because of exoosmosis, the cell shrinks. And when the cell shrinks, the content of the cell along with plasma membrane get separated from the cell wall. Or you can say, when a living plant cell is kept in a hypertonic solution, there develops a gap between the cell wall and plasma membrane, as plasma membrane gets detached from the cell wall because of the phenomena known as exoosmosis. And due to same reason, this phenomena which is occurring here is known as plasmolysis. Plasmo means plasma, lysis means breaking down or separation. So when plasma membrane is separated from the cell wall because of the process of plasmolysis or because of the process of exoosmosis is known as plasmolysis, right? So now this is all about plasmolysis, and now we talk about the deplasmolysis. Now we take a hypotonic solution. In this hypotonic solution, we have kept a plasmolyzed plant cell which has already undergone the process of plasmolysis, right? Now what happens when this plasmolyzed cell is kept in hypotonic solution? The water will enter from outside to inside, and here the process occurring is known as osmosis, because of which the cell swells and it regains its original position or original condition. And this process of regaining the original condition by a plasmolyzed cell when kept in a hypotonic solution, that phenomena is known as deplasmolysis, right? So this plasmolysis and deplasmolysis phenomenon could have been possible only because of the cell wall, right? So here, how can we define plasmolysis? Plasmolysis is separation of plasma membrane from cell wall when a living plant cell is kept in hypertonic solution. So that separation is known as plasmolysis. And now if we talk about deplasmolysis is so how we define deplasmolysis? That when a plasmolyzed plant cell is kept in hypotonic solution, it regains its original state due to endoosmosis, and that process is known as deplasmolysis, right? So that is all about the structure and function of the cell wall. And next component of the cell, which is a very important component, just as we have already said that it is the main controlling center of the cell, that is nucleus. So about nucleus we will be discussing in our next lecture. Goodbye till then, have a nice time.